Hello friends, welcome to Divya Mahesh tutorial. So in the last class, we studied DNA replication in prokaryotes, and today we'll focus on regulation of prokaryotic DNA replication. So in regulation of prokaryotic DNA replication, DNA ATP and ORIC, they both play a very important role. This was our initiation complex and this was initiation site. So first we will see initiation complex, how it plays a major role in regulation of prokaryotic DNA replication. So DNA ATP in its this form, adenosine triphosphate form, it is activated form and thus promotes replication. But in the presence of enzyme RIDA, that is regulatory inactivation of DNA A. In the presence of regulatory inactivation of DNA A, this complex inactivates and thus DNA ADP. And this DNA ADP inhibits replication process. So we saw DNA ATP. जब हम लोग प्रोकैरियोटिक रेप्लिकेशन पढ़ रहे थे तब हम लोग ने DNA ATP को इनिशिएशन कॉम्प्लेक्स माना था, जो कि आके बैठ रहा था और इनिशिएशन करवा रहा था, बाकी सब एंजाइम्स एंड फर्दरमोर एंजाइम प्रोटीन्स आके बैठ रहे थे. तो यही DNA ATP जो था ATP के फॉर्म में, एडिनोसिन ट्राइफॉस्फेट के फॉर्म में, एक्टिवेटेड फॉर्म में था और प्रमोट कर रहा था DNA रेप्लिकेशन को. बट इन द प्रेजेंस ऑफ एंजाइम रेगुलेटेड इनएक्टिवेशन ऑफ DNA A, ये किस फॉर्म में चेंज हो गया? DNA ADP डाइफॉस्फेट के फॉर्म में, ADP के फॉर्म में चेंज हो गया, जो कि इनएक्टिवेटेड कॉम्प्लेक्स है, जो कि प्रमोट नहीं करेगा DNA रेप्लिकेशन को. सो व्हेन सेल वांट्स रेप्लिकेशन, then this enzyme will be inhibited, and thus if this complex is formed before, अगर सेल पहले नहीं चाहता था रेप्लिकेशन तो वो इस एंजाइम को एक्टिवेट करके DNA ADP को बना के रख चुका होगा. पर अगर उसके बाद उसको रेप्लिकेशन करवाना है, then another enzymes come which makes the inactivated complex again to activate it, that is DOS. So we'll see that is DNA ATP was first inactivated to DNA ADP by RI, regulatory, will write full form here. This regulatory inactivation of DNA A. Okay, so in the presence of RIDA, this DNA ATP was inactivated to DNA ADP, and thus this will inhibit replication. Now this DNA A again activated in the presence of DARS, that is DNA A reactivating sequences, that is DNA reactivating sequences. So DNA A reactivating sequences will again activate this DNA ATP, and thus this will promote replication. So it depends on the cell when it is going to activate the RIDA and when it is going to activate the DARS. So this was the regulation of prokaryotic DNA replication by DNA AATP complex, that was initiation complex. Now we'll focus on ORIC, that is initiation site. ORIC. This site has 11 copies of GATC 5' to 3' sequences, guanine, adenine, thymine, cytosine. So 11 copies of this. So here enzyme works DAM methylase. So here enzyme works DAM methylase. We'll see its role in two prospects, that is first replication regulation and repair mechanism. From the name, it's DNA adenine methylation. So it will methylate adenine in the GATC sequences. Thus we'll see that first we'll see that if cell wants replication is, first we'll see the role in its repair mechanism. So first DNA replication has performed and it's over. Now it has to search the mismatched paired or unwanted pairs or wrong pairs in the sister strand, daughter strands. So it should make difference between what? Template and daughter strand. So in DNA replication, the template strands were methylated, daughter strands were not methylated. So they are formed like this. If it's parent and they formed like this, one parent and one daughter, one parent and one daughter. So this strand is methylated. Sorry, we'll see your daughter and will be a parent, otherwise it will be confused. So this is methylated. So this condition is our hemimethylated. Methylated DNA strand. So the enzyme will recognize the hemimethylated DNA and will attach and will cleave the daughter strands mismatched pairs. So methylation helps in repair mechanism through this process. Now what happens next is activity of DAM methylase, DNA activation methylation enzyme. It adds methylation to the adenine. We'll write here, that is sequences GATC 3' to 5'. So what DAM methylase does add methylates the adenine group of GATC sequences. So in our old strand, parent strand, this strand was methylated, but newly synthesized strands were not methylated. So when replication is completed and our daughter strands are formed, they remain unmethylated for some times, but soon they get methylated so that they could not be cleaved or again start the replication, because once in a cycle the DNA replication occurs one time. To ensure that the replication occurs once in one cycle of the cell division in prokaryotes, this should be methylated. So methylated DNA does not undergo replication. So this was the role of ORIC and DNA complex, ATP complex, in the regulation of DNA replication. Hope you understood. Thank you.